Assalamu alaikum. Today's lecture topic is protein turnover and nitrogen balance. Protein turnover results from simultaneous synthesis and degradation of protein. Under all normal circumstances, some proteins are continuously degraded, but they are replaced by circulating amino acid pool. So this is all related with the protein turnover. We will discuss in detail. And the nitrogen balance. When we use the term nitrogen, it means it is related with the protein. So in fact, that is the protein balance. Protein balance when the intake of protein is equal to the proteins excreted in different forms in the urine or stool. So when there is a balance between the two, that is called nitrogen balance. And we will study in this lecture if the intake of protein is more than the output, the result will be the person will gain weight, and that is called positive nitrogen balance. And opposite to that, if the input is less than the output of the proteins in the form of different compounds in the stool or in the urine, that will be a negative nitrogen balance, and in this condition the person will lose weight. Now, unlike fats and carbohydrates, amino acids are not stored by the body. Any amino acids in excess of biosynthetic needs of the cell are rapidly degraded. They are degraded in two phases. The first phase of catabolism is transamination and subsequent oxidative deamination. We studied in our previous lectures regarding the metabolism of amino acids, which result in the formation of alpha keto acid and free ammonia. This free ammonia is excreted. Primarily, the major portion of free ammonia, it is excreted in urine in the form of urea, which is synthesized in the liver, in the hepatocytes by urea cycle. So this is the first phase of catabolism, the release of free ammonia. The second phase of catabolism is the carbon skeletons of the alpha keto acids are converted to common intermediates of energy producing metabolic pathway. So this alpha keto acid skeleton is either it is utilized to form fatty acids, to form further amino acids, to form the glucose, or it is completely oxidized to be used as a energy source. This is the diagram which is clearly showing the whole protein turnover in our body. This is the amino acid pool, which is circulating in the blood, and some amino acids are present within the cell. Amino acid pool is contributed by three factors: dietary protein, which we are taking in our diet; body protein degradation. I told you, under all circumstances, some of the body proteins are catabolized. So after those proteins are catabolized, they are sliced into the formation of amino acid, and that a pool of amino acids come to this circulating amino acid pool. And synthesis of non-essential amino acids. Amino acids which are synthesized from essential amino acids which we take in our balanced diet, they also be added into this pool. So there are three factors which are contributing to constitute this amino acid pool. This pool comprises just 90 grams of total 90 grams of amino acids are present, and this has a central importance in regulating all the protein metabolism in our body. Now this amino acid pool, this is the input of amino acid pool, and these are the three possible outputs of amino acid pool. This amino acid pool may be used for the body protein synthesis like plasma proteins, enzymes, antibodies and muscle. Or this amino acid pool may be used for the biosynthesis of non-protein nitrogenous tissues constituents, for example porphyrin, creatine, hormones, neurotransmitters, purines, pyrimidines, niacin, thyroxine. So these are the non-nitrogenous tissue constituents, and this is the body protein synthesis, non-protein nitrogenous tissue constituents. Now excess, if we take excess protein in our diet, excess protein is catabolized, and after the catabolism there are three fates. Either it is used for the synthesis of glucose or stored as a glycogen, or it is used for the formation of fatty acids or ketone bodies, like we call those amino acids ketogenic amino acids, and these are the glucogenic amino acids. So some amino acids have only this property, glucogenic properties, some has ketogenic properties, and some amino acids has both these properties, ketogenic as well as glucogenic. Now the remaining excess, it is completely oxidized, or after donating its nitrogen, it is completely oxidized to CO2 or water to deliver energy. This is the urea formation in when excess of nitrogen, that is moved from amino acid, and in the liver that free ammonia is converted into urea. So this is the whole fate of the protein turnover. Input, 3 output, and excessive amino acid pool also has 3 fate. So I'm sure you will remember this diagram. Now the nitrogen balance. Most proteins contain 16% of nitrogen. This provides an accurate index to calculate the amount of proteins which are present in our food or which are present in our excreta, in stools or urine. So nitrogen content is determined chemically and multiplied by 6.25 factor to get the amount of protein present in the food and excreta. The ratio of nitrogen intake and output is called nitrogen balance. Output of nitrogen is in the form of urine and feces. When we use the term nitrogen, it means protein. The output of protein in the form of nitrogenous compounds in the urine or feces. If input of nitrogen or if input of protein is greater than the output, it is called positive nitrogen balance. That is, anabolism is high than catabolism, like growing children and pregnancy. So these children and pregnant women, they are in a state of positive nitrogen balance because they are gaining weight. Their input is greater than the output. If input of nitrogen is equal to the output, it is called nitrogen equilibrium. If input of the nitrogen is less than the output due to more catabolism, it is called negative nitrogen balance. For example, chronic wasting disease. So when we see any person, if a person is gaining weight, we can say that that person is in a state of positive nitrogen balance, and if a person is losing weight, so that will be in a state of negative nitrogen balance. Protein and its quality plays an important role to maintain normal nitrogen equilibrium. Negative nitrogen balance can be converted to positive when missing amino acids are added in the diet. Maintenance of body weight is also used an index for positive or negative nitrogen balance, or whether the person is in a state of nitrogen equilibrium, if he or she is maintaining their weight. Now, overall nitrogen metabolism. The role of body proteins in maintaining nitrogen balance involves two important concepts: amino acid pool and protein turnover. These two factors we will discuss in detail. Now, amino acid pool first. Free amino acids are present throughout the body, for example, in the cells, blood, and extracellular fluid are entertained as a single entity. They are amino acid pool. Input of the pool, as I showed you in the picture, there are three inputs for this amino acid pool. One, amino acids by degradation of body proteins, which is a normal mechanism. Number two, this is important contributor of this amino acid pool, the amino acids derived from dietary protein. And number three, the synthesis of non-essential amino acids, which when essential amino acids, they are metabolized in a different ways to form these 10 non-essential amino acids. So those 10 non-essential amino acids, they also are added to this amino acid pool. Now this picture shows, it is present in your Lippincott turnover. This is the turn, body protein. Under all circumstances, 400 grams body proteins are degraded per day, and definitely under these normal circumstances, this will have to be replaced. I will show you in another diagram where this amino acid pool is being used to form the same amount of body protein which has been catabolized. So protein turnover results from the simultaneous synthesis and degradation of protein molecule. In healthy fed adults, the total amount of protein in the body remains constant. So if 400 grams of protein is catabolized, at the same time 400 grams of protein will be synthesized in just sufficient to replace the protein that is degraded. Now the second important component of this amino acid pool is dietary protein. Dietary protein varies from person to person, from country to country. And the third one, synthesis of non-essential amino acids from essential amino acids which are present in our diet, so they are being added into the amino acid pool. So these are the amino acid pool's constituents, contributors, donators. Output of the pool: synthesis of body protein. There are three output, same as the input of the pool. Three input of the pool, three output of the pool. Synthesis of body protein, that body protein which already has been catabolized, degraded, so that has to be replaced. So that is contributed by this amino acid pool. Amino acids consumed as precursors of essential non-protein nitrogenous compounds. So non-protein nitrogenous compounds, they are being synthesized from the amino acid pool, creatine, pyrimidines, purines, thyroxine, like all those compounds. Or the essential amino acids which are present in the pool, they are converted, they are used for the conversion of amino acids to glucose, glycogen, fatty acids, or CO2. So either this pool is used for the formation of glucose to be stored at glycogen, or the formation of fatty acids or ketone bodies, or they are completely oxidized to deliver energy. So these are the two possible platforms: input of the amino acid pool and output of the amino acid pool. Now you can see here in this diagram the output of amino acid pool. This is the protein which was normally degraded per day, the same amount, 400 grams per day, that is being replaced. So it is contributed by the amino acid pool. This is one fate. Second, the 30 grams of per day, these non-protein nitrogenous compounds are synthesized. These long list of these compounds, porphyrins, 30 grams. Or this amino acid pool may be used for the synthesis of glucose, or may be used for the synthesis of fatty acids, or it is completely oxidized to CO2. The amino acids not used in the biosynthetic reactions are burned as a fuel. So excess amino acids come to this track of providing the fuel to the body. The overall nitrogen balance depends upon two factors: amino acid pool, which we discussed in our previous slides, and the protein turnover. Protein turnover, most proteins in our body are constantly being synthesized and then degraded, permitting the removal of abnormal or unneeded proteins. Some of the proteins are rapidly degraded and at the same time resynthesized to maintain the normal amount. Some of the proteins remain fairly constant, like collagen, because their levels are maintained by selective degradation. So net amount of proteins present in the body depends upon further two factors: the rate of turnover and protein degradation. How much protein is degraded and in which time that protein is being replaced. These are the two factors which are related with the protein turnover, that is rate of turnover and protein degradation. Now, rate of turnover. In healthy adults, the total amount of protein in the body remains constant. The rate of protein turnover varies widely for individual proteins. For example, short-lived proteins, for example, regulatory proteins and misfolded proteins are rapidly degraded, having half-lives measured in minutes or hours, while long-lived proteins with half-lives of days or weeks constitute the majority of the proteins in the cell. The structural proteins such as collagen are metabolically stable and have half-lives measured in months or years. Now, protein degradation. Two major enzyme systems responsible for degrading damaged or unneeded proteins. First is the energy dependent, and the second one is non-energy dependent. The energy dependent mechanism is related with the Ubiquitin-proteasome mechanism. Proteasomes mainly degrade endogenous proteins. This is important point to remember. Where energy is used, Ubiquitin-proteasome degrade endogenous proteins, that is, the proteins that were synthesized within the cell. And non-energy dependent degradative enzymes of lysosomes, no energy is required for this type of degradation. Lysosomal enzymes degrade primarily extracellular proteins such as plasma proteins that are taken into the cell by endocytosis. So there are two mechanisms. One is energy dependent for endogenous proteins, and one is non-energy dependent for the extracellular proteins. Now this picture is showing how the endogenous proteins are degraded by Ubiquitin-proteasome mechanism. Proteins selected for degradation are first covalently attached. Remember, this is the covalent attachment to ubiquitin. These are the ubiquitin molecules. They tag the cellular proteins. A small globular protein, these are the small globular protein, ubiquitin. The consecutive addition of ubiquitin moieties generates a polyubiquitin chain like this, tandemly linked एक दूसरे के ऊपर molecules of ubiquitin. So this is the target protein. It is tagged with ubiquitin covalently, and the process is called ubiquitination. Ubiquitination of the target proteins occur through covalent linkage of alpha carboxylic glycine of ubiquitin, alpha carboxylic group of ubiquitin. It is covalently attached to the lysine amino group on protein substrate. So on protein substrate, amino group of lysine is covalently attached with alpha carboxylic group of glycine, covalently attached. This protein ubiquitin complex, this complex is recognized by a barrel-shaped macromolecular proteolytic complex called proteasome. This is a proteasome which functions as garbage disposal. The proteasome cuts the target proteins into fragments. This protein is cut into fragments which are further degraded into amino acids which enter the amino acid pool. So here you see protein selected for degradation is tagged with molecules of ubiquitin covalently attached. Second, this one, ubiquinated protein, and the process is called ubiquitination. This ubiquinated protein are recognized by the cytosolic proteasome, which unfolds and transports the protein to the proteolytic core. And here in the center of the core, this target protein is degraded into its different polypeptides or dipeptides, which are further finally degraded into monomers of amino acids. The proteasome cuts the target protein into fragments. This proteasome cuts the protein into fragments that are then further degraded into amino acids. This you can see. It is having a non-specific proteases which cut the proteins at all the different peptide bonds, leading to the formation of amino acids, which enter the amino acid pool. Now these amino acids will enter the amino acid pool. The ubiquitins are recycled. So after passing through this proteasome, proteins, unwanted, unneeded protein is degraded finally into the amino acids, and ubiquitin are set free. Globular proteins are set free, and it provides a shuttle-like service. Once again, it again attaches with the unneeded, unfolded protein and degrade that protein, tag that proteins to go for its cycling in the proteasome. Peptide fragments produced by the proteasome are degraded into amino acids. Proteasome ubiquitin complex requires ATP. Here, this attachment of ubiquitin to the protein, it is the energy dependent reaction when the ubiquitin tandemly attaches with this protein covalently. Now, chemical signaling for protein degradation. A protein degradation cannot be random, but rather it is influenced by some structural aspects of the protein. For example, some proteins that have been chemically altered by oxidation or tagged with ubiquitin are preferentially degraded. So altered protein, unfolded protein, a damaged protein, they are degraded first. They are immediately tagged with the ubiquitin. The other factor is the half-life of the protein is influenced by the nature of N-terminal residue, that is, which amino acid is attached on the N-terminal. If on the N-terminal it is serine, the half-life of that particular protein will be 20 hours. Similarly, if the N-terminal residue is aspartate, the half-life it becomes shorter, that is 3 minutes. Similarly, proteins rich in sequences containing proline, glutamate, serine, threonine are rapidly degraded, so they have short intracellular half-lives. Thank you very much. Thank you very much for today's lecture. We'll meet in our next lecture again. Stay safe, stay blessed.